This is a monthly program sponsored by Cohen Veterans Bioscience for the brain research and systems biology communities. Cohen Veterans Bioscience is a nonprofit research alliance whose mission is to transform research in veterans' mental health through translational research. We are focused on understanding the molecular underpinnings of diseases such as PTSD and TBI to develop better treatments and diagnostics. Before I introduce today's speaker, let me walk you through some logistics. This program is being recorded and will be archived in the Cohen Veterans Bioscience YouTube channel and on the Cohen Veteran Bioscience website. The presentation will be approximately 45 minutes in length to allow for Q&A afterwards. Please use the Q&A feature on the website to post a question, and we will pose the question to the presenter. Today, our speakers are Drs. Mony J and Silvia Fossati. Dr. De Leon completed his doctoral work in gerontology at Columbia University in 1980, followed by 2 years of NIH postdoc training in CT brain imaging at NYU and PET imaging at the Brookhaven National Laboratory. Dr. De Leon's scientific goals have consistently been to develop and improve early diagnostic accuracy and mechanistic understanding of Alzheimer's disease and traumatic brain injury. He has published over 400 papers and sits on many national and international scientific review boards. Dr. Silvia Fossati is Assistant Professor of Psychiatry and Neurology at the NYU School of Medicine and the Director of the Biofluid Biomarkers Core at the Cohen Center at NYU Langone Medical Center. She is committed to the understanding of the molecular pathogenesis of neurodegenerative and psychiatric disorders. She obtained a PhD in molecular and cellular neuropharmacology at the University of Florence, Italy. She joined the NYU School of Medicine as a postdoc fellow in 2007 to conduct research focused on understanding signal pathways that trigger cell toxicity and cellular stress responses in Alzheimer's disease and cerebral amyloid angiopathy, with a focus on mitochondrial dysfunction, deregulation of apoptotic factors, neuroinflammation, and stress of cells composing the neurovascular unit. In 2015, she received an appointment as assistant professor in the departments of neurology and psychiatry, thereby establishing her independent laboratory. I am pleased to present Drs. De Leon and Fossati, who will speak on the subject of evaluating neurodegeneration following TBI, some new ideas. Dr. De Leon.
Good afternoon, everybody. This is Mony J. I'm here with Silvia Fossati, and we welcome you. We have my disclosures here. Let me begin by saying a few words about traumatic brain injury itself. Worldwide, this is a problem that affects some 10 million people annually. Currently, we define traumatic brain injury by the response of the individual, meaning that we really don't have behavioral response, meaning we really don't have biological markers to really characterize and fully understand what the brain is experiencing. Among people that have traumatic brain injury, some of the symptoms include impaired sleep, difficulties in attention, learning, and memory. Of course, and these are very dose-dependent types of phenomena that occur following these injuries. Now, traumatic brain injury disproportionately affects children. We know about, from the media currently, there's great importance in athletes that are being traumatized. And of course, our military veterans for a long time have been a topic of great concern to all of us. Now, repeated injuries increase the risk for dementia. And let me show you some data that we've put together. This was done by one of my students who shows that the age of onset of dementia is about 10 years earlier when you have a history of multiple traumatic brain injuries. Individuals that have a single traumatic brain injury, that's the line in the middle, also have an advanced age of onset. But this is a very important observation done on a data set that had nearly 14,000 subjects. Now, as multiple traumatic brain injuries appear to be linked to an endpoint of chronic traumatic encephalopathy, therein lies the tip of the iceberg in terms of the aggressiveness of the symptoms and the features and the rapidity of onset. These individuals that have this kind of profile clinically are marked by aggressive behavior, fluctuating consciousness, dementia, and it's often associated with substance abuse. Now, in the pathology of CTE, neurofibrillary pathology, NFT, is very salient. There was a large consensus meeting that was recently published that showed there were changes that were quite systematically observed: tauopathy in the medial temporal lobe, in the superficial cortex, and in the depths of sulci and blood vessels. And so neurofibrillary pathology is a key feature of CTE. What you see here is a data set from Barrios and company at UCLA that applied an imaging agent using the positron emission tomography technique. The technique that they've used, FDDNP, is a marker for both tau and amyloid. It's not selective for tau. Nevertheless, in regions where tau does accumulate, such as in amygdala and hippocampal formation, and amyloid is relatively resistant to accumulating in those areas, these scans are showing that individuals with CTE are in fact having an increased signal intensity marked by those red blobs in regions that are typically vulnerable for tau pathology. Histologically, our group has developed some technology which we think may bear on this quite interestingly. This is a distribution of amyloid and tau pathology from the PET scan on the top row, and from the histology, from the post-mortem observations on the bottom row. And basically the lesson that you see from this slice going through the temporal lobe, parahippocampal gyrus, hippocampus, amygdala, and of course some of the cortical areas to the far left of the pictures, there's a different distribution between amyloid and tau. Amyloid is very broadly distributed throughout the cortical ribbon, whereas the tau pathology in Alzheimer's disease is much closer to the white matter. It's much more deep. On the other hand, the reports on CTE are very consistently showing that there's a superficial involvement of tau as well. So we have developed this strategy, this protocol, for actually distinguishing the laminar distribution of these types of lesions. And although this has not been done yet, we're actually actively collecting tau data from individuals with CTE and traumatic brain injuries to see if, in fact, we can distinguish the laminar distribution between the deep tau pathology of Alzheimer's disease from the superficial tau pathology of traumatic brain injury. As I mentioned a moment ago, the blood vessels are uniquely involved with tau pathology in CTE. And you see here these black dots surrounding the vessels, which are in fact the amyloid, the tau pathology. This is work that comes from Blenau's research. Now, many patients, not all, but many patients with CTE have elevated amyloid levels in the brain. It's been estimated that as many as 40% will have this feature, and it tends to be those that are more severely impaired, which is really kind of interesting because it's not Alzheimer's disease. Alzheimer's disease basically is defined by the presence of amyloid and tau pathology. So this means that CTE patients, while the majority of them do not meet criteria for Alzheimer's disease because of the absence of the tau pathology. What's very interesting in this question is the time course of amyloid pathology. If you look during life, and then these are now PET scans looking at traumatic brain injury subjects, and the scan is identifying the amyloid distributions in the brain. If you look at the healthy person example at the very top row, you see there's some uptake in the middle of the brain. It's largely confined to the thalamus and to the white matter and brainstem. The second row shows an individual with traumatic brain injury, also a young person, but now you can see the spread of the amyloid into the extremes of the brain, into the cortex of the brain. But if you look at somebody who was studied virtually 2 months later, you see that there's much less amyloid. And if you look at somebody who was studied almost a year later, there's even less amyloid. So it looks like the time course, it's suggestive. It's not proof at this point because we don't have that observation. But the time course suggests that there's an early amyloidosis that then is somehow cleared. Now, this view is consistent with what is seen more graphically in animal data. Animal studies are showing that there's this course of amyloidosis that is a relatively early phenomenon and then wanes. Inflammation is a very important feature of tauopathy, of tauopathy, amyloid and amyloid pathology. And in the case of CTE and TBI, inflammation is a standard feature. Here what you see is another PET scan highlighting the capacity to measure some aspect of the inflammatory response using a PET image. The bottom image shows more uptake in a football player, former football retired player, as compared to an age-matched control. So there is an inflammatory response in addition to the lesions that can be detected in association with TBI and CTE. Now, part of the characteristics of TBI and CTE, which has been fairly well recognized in animal studies, is the damage to the blood-brain barrier. This is a very important thing because molecules that are of a certain size and characteristics are not necessarily supposed to get in, and other molecules are not supposed to necessarily get out as quickly as they might. The concept of in vivo studies of blood-brain barrier in human has been rather difficult to discern. The technologies haven't been as sensitive, and for minor injuries, it's been a challenge to actually get images that are informative. There's a new technology that uses MRI called DCE MRI. It's a dynamic contrast enhanced MRI, which basically injects a gadolinium type marker for blood-brain barrier integrity, but it's done so in the context of collecting dynamic data, which is a technique that's used very often in PET scans. So it's a combination of using dynamic input of information to actually sensitively mark the time course of acquisition of voxels in the brain that are seeing this contrast agent. And in this work that was published just recently by Zlokovic and company by Montan, you see that the hippocampus is vulnerable in Alzheimer's disease, whereas other parts of the Alzheimer brain were not. This type of work needs to be applied to the TBI-CTE complex. So finally, in terms of the pathologies I wanted to mention, and also to spend a little bit of time with, is the so-called glymphatic clearance. Now, glymphatic refers to the combination of glial and lymphatic tissues to actually facilitate the clearance of spinal fluid from the brain. Spinal fluid is known to be produced in the choroid plexus of the brain. It comes from the blood, and then it's cleared. It's actually turned over about 3 times every day, 3 to 4 times a day. And the black marks here on this image show you the ventricle in the center, which is a fluid-containing vault of CSF. It shows also around the brain the subarachnoid distributions of CSF, and you see these streams of CSF going into the spinal cord on your right and coming out of nerve fibers on the left. These are the presumed pathways for spinal fluid removal. At a more molecular level, the spinal fluid is actually delivered from an arterial source. It passes through the brain. I'm sure this is a great shortcut of description. It ultimately ends up in the interstitial fluid of the brain surrounding nerve fibers, and then is picked up by perivascular channels in the venous architecture and delivered both to subarachnoid space and to the venous compartment for removal. And so the waste products of the brain are caught up in these interstitial fluid motions, and they themselves are flushed into the perivascular channel and then filtered out and removed from the body. When you look more closely at this, you find that in diseases like Alzheimer's disease, where clearance is suspected as being abnormally slow, bad guys like amyloid beta accumulate in these paravascular channels, and you see here on the bottom left in red the bad guy in the channel, and this clogs the passageway, making the efflux of fluids more difficult. It's presumed that this is part of the basic mechanism by which amyloid begins to deposit in the brain because of the slowness of its clearance from the brain. Here you see work by Carrera, which is this is basically an anatomical study. What Roxana had done here is actually label the amyloid and then inject it into mouse brain, and you can see here in pink the areas where the amyloid actually goes. So this is using the natural amyloid beta as a tracer by labeling it and then plotting its paravascular distribution. So this very elegantly shows you the way the garbage is basically removed in these paravascular channels. Now, if you look more globally at the efficacy of removal of spinal fluid, with age, there's a decrease in the capacity of the system to remove fluids. There's a reduction in spinal fluid production. There's a reduction, obviously, in spinal fluid clearance because they're linked. It's a homeostatic mechanism. But there's an even greater deficit channel-wise that can be detected in aging brain. And so the image on the right is basically an old mouse. But the next image is very interesting because this is now traumatic brain injury. And you'll see here in the image on the right, looks very much like the old mouse that I just showed you. So here you have a young mouse that has had an injury, and these channels are somehow no longer performing as efficiently as they were, and the egress of these labeled tracers has been reduced dramatically throughout the brain. Now, one of the ways in which these channels is affected and has been recently recognized mechanistically through the work of Nedergaard and her associates, Jeff Iliff for one, has been to this relationship between sleep and clearance. It appears, and we've seen this in the animal models now quite routinely, not yet fully translated to the human, but likely to be in a similar direction, that during sleep there is an increased clearance of these molecules. And this is facilitated through proteins like aquaporin that facilitate the water movement from the brain. These are astrocytic proteins that basically shrink and enlarge the spaces, the channels. So at night, the channel is expanded. The volume of fluids that can run through the paravascular channel is increased. If you decrease the sleep, if you impair the sleep experimentally, you reduce the amount of efflux of the tracer, of the marker. And so it raises this question: in sleep disturbances, are we in fact damaging the brain through a process of the failed clearance of byproducts of metabolism and waste products from the brain? On the right, I put in red there some observations that we made from our own populations here at NYU, that individuals, normal individuals, that have a history of traumatic brain injury are 4 times more likely to have impaired sleep. Now, of course, we don't know the chicken and the egg relationship here, but we know that traumatic brain injuries can cause poor sleep. We know that traumatic brain injuries are affecting clearance, and we know that sleep is affecting clearance. Is it a double whammy, or and what is the actual chronological relationship between traumatic brain injury, sleep, and clearance dysfunction remains to be revealed. Now, patients with traumatic brain injury, also from our data, are really now coming under active investigation. Let me show you a few summaries of what we know about human clearance of fluids from the brain. If you take a PET scan, here being an example of a tau scan, if you look at the pictures, you'll see that there's more tau in the brain of the Alzheimer patient. If you look at the green line in the graph, you'll see, relative to the red line, you'll see that the blood, which is red, rapidly clears. So when you measure the blood that's in the brain, the tracer that's in the blood goes away quickly. The brain is much more slowly removing contrast agents. And so basically about 75% of the tracer that is delivered to the body, it gets to the brain and is removed from the brain, making the clearance of tracer important and a target for investigation. So what we've done is in blue here, we made a mask, so to speak, of all the parts of the head that were not including the brain. And so you have the blue non-brain and then the brain. If you look at the non-brain part and you actually measure the clearance of the radio tracer, what you find is that there's a very strong signal that appears in the area of the turbinates of the nose, the superior turbinates, just where the cribriform plate is, which is that plate under the frontal lobe of the brain where the olfactory nerves pass through. And in the animal models, this is a consistent feature across all mammals that has not been recognized before in the human. It's not been demonstrated before in a living human. So these are the first images of the functional capacity of the turbinate area to clear the radio tracer from the extra brain compartment. You'll notice that there's not a lot of tracer being removed anywhere else in that area that was out of the brain. It's largely centered in that area. That doesn't mean that there aren't areas other than this. They may well be below the resolution capacity of the PET instrumentation. But PET can give you also an image of the brain itself. And so here you see, this is now a clearance image. This is now the failed clearance in the brain of Alzheimer patient on the right compared to the more robust clearance in a normal subject in the brain. So what we did was to look more in depth at this relationship between disease clearance and brain lesions. We took an area in the brain which is vulnerable to amyloidosis, even in a normal brain. And so we looked at normal elderly subjects. We looked at many subjects, but the target here, initially what I'm describing is on the normal subjects. The anatomy is on the far left. The cingulate gyrus target is in green. We mapped basically the top and bottom third of all the clearance on top of the green. So if it's in blue, it means that it's lower clearance for the normal distribution. It's a normal distribution, it's the bottom third. And the top third of the distribution is in yellow. And you can see the middle distribution still remains green. So we're mapping high and low clearance on top of this anatomy. We then mapped the amyloid that's found in those brains, in those normal brains, and that's in red. And you see the amyloid with respect to the anatomy. The amyloid red is a small fraction of the total anatomical size of the cingulate gyrus. Then we mapped the low clearance and the high clearance on top of the amyloid, meaning that the blue and the yellow are mapped on top of the red. And you can see here that the blue covers almost all the red, meaning that clearance is very highly associated with amyloid. And on the graph below it, you'll see for all the groups, basically, you'll see the same phenomenon, that if you have amyloid in the brain, the clearance is lower. And the clearance is measured with a different technique altogether. The clearance is measured on the tau scan. The amyloid is measured on an amyloid scan, here called PIB. So, trying to understand these markers with respect to clearance, one has to also consider the production of molecules that are being cleared and the deposition. And they don't always work in the same direction. When you have a clearance problem for CSF, it means that the molecule has already passed through the membrane. And when there are clearance deficits in the flow of these molecules out, when they're in the CSF, it tends to raise the levels. On the other hand, when you have deposition of these proteins in the brain, the amount that's cleared to the CSF is less. Therefore, you should have reductions in the levels. And as such, the time course is very informative. If you look at plasma A beta in traumatic brain injury, this is now on the x-axis days after in the level expressed, basically considered a marker for injury. If you look at total tau, you'll see on the far left a spiking of the levels in the blood, and then a return to baseline as the individual recovers. If you look at the neurofilament light chain protein, another MAP2 protein like the tau is, you see also an elevation, a bit delayed as compared to tau, but you also see an elevation. So it raises the point that you may have a reduced clearance in both AD and in TBI, and that this reduced clearance may actually contribute to the short-term elevation in the apparent levels of these analytes in the blood and possibly in the spinal fluid as well. Long-term, there are changes that are seen in deposition. So it means then that clearance as a potential target needs to be understood in the chronology of the clearance. So it's with great pleasure that I introduce my colleague Silvia Fossati, who I hope will give you a very much more erudite explanation than I've just done.
Thank you, Mony. So let's go here from the broad imaging point of view that Mony gave you to a more molecular and cellular point of view. And let's see what happens in the brain to these cells and to these molecules after injury due to TBI. So we have two main episodes, which are the axonal damage and the vascular damage, which are both due to mechanical stress and shear stress due to the injury. In the axons, we have molecules such as NFL and tau that are released from the microtubules due to injury, and also amyloid beta that is released from the neuronal cells, and both tau and A beta are prone to aggregation. So A beta will form oligomers, protofibrils, and then will deposit as fibers in plaques, and tau will be phosphorylated, and phosphorylated tau tends to aggregate and form tangles. There are also other molecules that are produced by glial cells after injury, which are cytokines, so inflammation molecules, and GFAP, which is a structural glial protein. Immediately after injury, there is a blood-brain barrier dysfunction. So some of these molecules that are produced acutely will go to the blood and will be visible as biomarkers, as our lab and others have done after TBI. But what happens more chronically? So some of these molecules that are aggregating, such as amyloid beta and tau, will aggregate around and on the surface of the blood vessels. And this aggregation process is known to induce toxicity on the blood vessels. So chronically, there will be an enhanced toxicity, and there will be perivascular damage with endothelial cell death, cytochrome C release from the mitochondria, and this will induce blood-brain barrier permeability also over time. And other molecules that are part of the blood-brain barrier or structural molecules of endothelial cells will be released in the blood, for example, junction proteins, MMPs, and others. So if you look over time at these blood biomarkers and at biomarkers of vascular dysfunction and clearance dysfunction, these will likely be correlated, and they will give us a full picture of what happens in the brain after TBI. To understand how these cells in the brain interplay with each other, it's very important to know what the neurovascular unit is. So what happens when we have this amyloid around the vascular cells? And this is something that can happen after TBI, but it can also happen in disorders that affect the production, the deposition, or the clearance of the peptide. We know that the vascular cells, as I was telling you, interact with each other in the neurovascular unit. So this deposition of amyloid can cause inflammation, interacting with glial cells, can cause hemorrhages in the vasculature, hypoxia that also affects the rest of the brain, including the neurons, and that will cause neurodegeneration. And we know that amyloid deposits around the vessels, which are in pink here, shown by 2-photon microscopy, affect vascular health, neurovascular health, and in particular the oligomers are the ones that participate in causing neurodegeneration. So what happened with the amyloid toxicity and with the amyloid aggregation? We have amyloid beta monomers that are in equilibrium with fibrillar deposits. And monomers and fibrils are in equilibrium with each other, but there are intermediate aggregation species, which are oligomers and protofibrils. These oligomers and protofibrils are known to be the toxic species from a lot of work, including work done in our lab. So the model that came out from the work done in my lab was that when amyloid aggregates around the vessels, forms oligomers and protofibrils, and those activate death receptors on the cell membrane in endothelial cells. The death receptors are increased, their expression is increased, they are activated, they induce caspase 8 activation, which will cause release of cytochrome C and of ROS, reactive oxygen species, which are correlated to aging and cell stress. And then caspase 8 and 9 will activate caspase 3 and induce cell death. So how can we stop these pathways? One way could be silencing the death receptors, which was efficient in our studies in vitro, but is a little harder to do in the brain as there are no specific inhibitors yet. The other way is to block the mitochondrial dysfunction pathway. And we've tried to do that, and we've been successful using repurposed drugs that are called carbonic anhydrase inhibitors. And I will tell you more about these. So why the mitochondria are so important? Mitochondria are a very important target for both traumatic brain injury and Alzheimer's disease therapy. First of all, because they are the energy source of the cells, so if they fail, the cells will lack energy and overall die. They are responsible for the production of free radicals, or ROS, reactive oxygen species, which induce cell stress and aging. And they also release cell death signals such as cytochrome C. So now, using these repurposed drugs, we were able to prevent both the production of free radicals and the release of cytochrome C. What are these drugs? They are FDA approved, called carbonic anhydrase inhibitors, and two of the ones we've used are methazolamide and acetazolamide, which are widely used clinically for glaucoma, as a diuretic, and for the prevention of high altitude sickness, among the other things. And they are also known to induce vasoreactivity and cerebral blood flow. We found that the mechanism of actions are multiple, and that they reduce the release of cytochrome C in cells challenged with amyloid beta. They also reduce the production of hydrogen peroxide specifically from the mitochondria, so reducing oxidative stress. And they reduce caspase activation and eventually cell death. And this is true for vascular, neuronal, and glial cells. If used in mice, they improved memory, mouse models of cerebral amyloidosis. And they are also able to reduce amyloid pathology in the same animal models. And this is probably correlated with the fact that they are also known to increase cerebral blood flow and vasoreactivity and reduce cerebral edema. Here are some experiments that we did in mice. First, we injected amyloid beta in the hippocampus of these mice. And as you can see, you have A beta in red on the left, and the active form of caspase 3, which is indicative of cell death pathways, is in green. And these are caspase 3 is activated in the same cells that have amyloid. And if you look at the whole hippocampus, the level of caspase 3 activation is increased with amyloid beta, but it's not increased in mice that received methazolamide intraperitoneally 1 hour before injection of amyloid beta. Also, if you look at the number of neuronal cells in the hippocampus of these injected mice, you see that the mice injected with A beta had less living neurons. But if the mice were previously given methazolamide, the number of neurons was more similar to the control. We recently did this also in transgenic mice, and this is a model of a vascular amyloidosis. So these transgenic mice with vascular amyloidosis had a lot of red, a lot of amyloid in the brain, as you can see in the second picture right? And they also had yellow, which means that caspase 3 was activated. But if you look at the mice that had acetazolamide and metazolamide, they have a lot less yellow, so a lot less active caspase, and definitely less red, which means that also the amyloid pathology was reduced. If you look at memory in these mice with Barnes maze, you see that, of course, the transgenic mice have their memory worsens with age, and at 15 months they have memory issues, so they take longer time to find the escape hole in the maze. Or longer distance in this case, measured by distance. And then when you give this metazolamide and acetazolamide to the mice for a few months before testing them, you see that the memory is very similar to the one of the control, so they improve memory. From a more in vitro point of view, let's look at what these peptides do to the blood-brain barrier. So you can put in this system endothelial cells on electrodes in some wells, and they will form a monolayer in about 48 hours and reach a plateau in resistance. At that point, you can give them peptides, amyloid peptides, and see what happens to the resistance. And in particular with A-beta-42 and Q22, which is the Dutch amyloid vasculotropic mutant, and these are peptides that are prone to aggregation and to forming fibrils, we have a drop in barrier resistance, basically. So the resistance of the blood-brain barrier is affected by these peptides. Not only, as Mony was mentioning, we know that we have a lot of inflammation in the brain after TBI, and in particular there are molecules such as interferon gamma, IL-6, IL-8, IL-1 alpha that are released from glial cells. So all these molecules affect the blood-brain barrier, and some more than others. So interferon and interleukin-1 alpha have more effect, for example, than IL-8. What does tau do? So in our lab, we have a diagnostic tau project, which means that we are studying how tau is released after TBI, after multiple TBI in Alzheimer's disease, and how much we can find in the blood. And actually recent studies show that tau alone can remodel vessel walls preceding amyloid deposition and may contribute to the downstream effects of amyloid on microvascular pathology. It has been also shown that tau can induce blood-brain barrier breakdown in animal models of tauopathy, so similarly to what we've shown for amyloid beta. Therefore, targeting tau may also be a strategy to limit vascular dysfunction and the associated neurovascular damage that is found in TBI, CTE, and Alzheimer's disease. And to conclude, I will tell you about some therapeutic approaches that can be used to reduce neurovascular dysfunction after TBI. Some are classical. There have been studies, for example, in Alzheimer's disease, and they can be the removal of tau and amyloid peptides by immunotherapy, which has given not so good results in clinical trials. So, and also we can reduce inflammation using, for example, dexamethasone, which has been shown in animal models to reduce chronic, dysfunction after TBI. But there are also some novel approaches that we are proposing, and those have to do more with affecting clearance and vascular health. So one could be a sleep therapy, and as Mony mentioned before, more sleep improves clearance. The other one is exercise, because you have an increased cerebral blood flow in the brain, so these as well could improve clearance. And there are some molecular mechanisms that could be used also to facilitate clearance, such as playing with aquaporin 4 that affects. Is known that the knockouts of aquaporin 4, for example, have reduced clearance. And then another strategy that we propose is to increase vasoreactivity and cerebral blood flow, and at the same time reduce mitochondrial dysfunction with the use of carbonic anhydrase inhibitors. So at this point, I give you back to Mony.
So thank you, Silvia, and thank you all for listening. So this is the time when, if you'd like, send us any questions, make any suggestions you'd like, and if it's not convenient for you online like this, do so offline. I'd like to point out that our studies have encompassed quite a number of groups and labs around the world, and so here is a list of our collaboration base, and we're very grateful for their continued participation. So while we're waiting for a question to come in, I'd like to ask Silvia a question. Silvia, what do you think, what's your vision and view of like looking at the timeline of post-traumatic brain injury? How would you describe the most important period to collect biological samples? Are we talking about very closely spaced samples in the immediate aftermath of an injury? Do we need to know something about long-term evolution of changes? There's a great uncertainty in this field. How would you optimize a study if you had really the resources that you need to do this right?
Yeah, great question, and it's actually a very relevant question, and I think that many people are asking each other this question. So for what we think, the molecular point of view I just gave you is that, okay, there is an acute damage, and at that point there is neuronal dysfunction, vascular dysfunction, and you find molecules in the blood, and that will last for probably a few days, 2 weeks, and then go back to normal over time. But what happens chronically, and what happens if the TBI is not one only, but there are more than one TBI? So this acute period eventually will try to go back, but then if you have another TBI event before it goes back completely, the system is already dysfunctional. So at that point, you will have a raise and an increased dysfunction that is higher compared to the levels of the first TBI. So basically, after the second, the system wants to go back to normal, but we can't really go back to normal completely, and this is even worse if you increase the number of TBIs. So what are the good time points? Well, there are early time points where you can see the injury, but there is, I think, a window in between the early time points and the chronic damage, which you can see in CTE or Alzheimer's disease later in age. And in that window, you probably don't see much from a biomarker standpoint, or at least not with the biomarkers that we have available right now. Maybe we need to find new biomarkers to look at that window. But you know, it would be very useful to look at what happens in the brain at that time, and gene expression and alteration of cell stress pathways in the brain. So I think we will need to probably look at different biomarkers for that window in time.
But what comes to mind to me, Silvia, in hearing your response, is that an injury occurs to an individual who his or herself is already experiencing changes related to age and reflecting their genetic background. So we know that so much of these phenomena that relate to the clearance of waste products are dependent on cardiovascular integrity. It's the heart and the pulsation of vessels that is actually driving the fluids out of the brain. And so individuals that come in with hypertension or individuals that develop hypertension may be at particular risk. It may be both a risk factor and a treatment target, looking at the input side of some of these disorders, and of course the genetics, which we haven't discussed at all today. Are there vulnerability genes? Are the same genes that make you vulnerable for Alzheimer's disease making you vulnerable for the deposition of similar pathology following TBI? It remains really unexplored.
Absolutely, and actually cardiovascular risk is very relevant, as we noticed that this is obviously one of our main targets, and we noticed that individuals that have cardiovascular risk without TBI or without Alzheimer's, they already have those biomarkers elevated, for example, amyloid beta and tau in blood, so that there is something that in those individuals makes it more difficult to go back to normal.
Yes, thank you, Silvia. Have any questions come in?
Yes, we do have one question. How does tau tracer measure glymphatic clearance?
Okay. The tau tracer, and it's not unique to the tau tracer, basically all tracers are cleared from the brain in this case. The question is how much is being cleared and at what speed. So an ideal tracer to measure glymphatic function in the brain would be a tracer that does not bind in the brain, but a tracer that has very good permeability. It gets into the brain. It gets out of the brain very rapidly. It has a low molecular weight and can be measured over a time course, so that's relatively long. So it has a label that on the order of, say, a carbon, which has a 20-minute half-life, or fluorine, which has a 118-minute half-life, would be probably optimal. Tracers that have very short half-lives, like oxygen, level 15 water, the half-life is 2 minutes, and it's probably not long enough to capture these views. So basically, the principle behind the measurement is sampling the brain and the brain's reduction in the concentration of the isotope after blood levels in the brain have returned to a baseline level, which, as you know, the blood is circulating very rapidly through the body, and the tracer is administered to the blood, it's delivered to the organs in the body, and then it's returned back to the blood and cleared. And the blood levels show this effect very rapidly. Within the first few minutes, they peak, and then they rapidly return to near baseline levels. After the return to baseline is a very good time for measuring the residual movement of tracers through the brain that's not confounded by the blood. So in answer to your question directly, it's not specific to a tau tracer, but a tau tracer in this case was used to make the measurement.
I don't see any more questions. So I think the best thing to do is say thank you for a wonderful and interesting, exciting presentation. We appreciate your time. And if we do get any other questions, we'll make sure to email them to you. Thank you so much.
Thank you.
Thank you, Evan.
Bye, everybody.
Bye-bye.